When we consume proteins in our diet, in order for those proteins to be used, our body must metabolize them first into individual amino acids and then ultimately reassemble them into the specific proteins that we need for life. The start of this process of breaking down proteins involves what is called denaturation, where denaturation is the focus of this particular video. Let's take a closer look. What do we mean when we say protein denaturation? Here is the structure of a so-called normal protein. That is referring to a protein that has all of its three-dimensional elements intact. In other words, it has the secondary structure of the alpha helices and beta sheets. It has the tertiary structure that is the three-dimensional shape of one single peptide chain. And if applicable, it also has the quaternary shape in place. Remembering that the quaternary shape refers to multiple proteins that are associated together to collaboratively work toward a specific function. Now, when we denature a protein, what happens is that the secondary, tertiary, and/or quaternary structure is disrupted to ultimately yield what's referred to as a denatured protein. So you can see here in this schematic that the normal three-dimensional shape of the protein has been completely disrupted. By disrupting the three-dimensional shape of a protein, the function of that protein is disrupted. Additionally, by denaturing a protein during the course of converting proteins into their individual amino acids during metabolism, when we denature the protein and disrupt that three-dimensional structure, this denatured protein structure is easier for ultimately enzymes to come in and act and break the peptide bonds within the protein in order to release individual amino acids. And we will get into that chemical reaction in the upcoming couple of videos that takes a denatured protein and breaks the amide linkages or peptide bonds within it to release individual amino acids. For now, we focus just on this process of protein denaturation. So what are some common ways that proteins can be denatured? Eggs provide us with one example that has a very nice visual representation, and that is when we look at an uncooked egg, such as this egg yolk shown here, compared to a scrambled egg shown here, there's a very clear difference in the texture of the cooked egg relative to the raw egg. And the reason is that heat is one key way that proteins are denatured. And the denatured protein seen here in the scrambled egg panel has ultimately a very different texture than the proteins that have their three-dimensional shapes intact in the raw egg. Another common way that proteins can be denatured is by treatment with acids, such as acetic acid or hydrochloric acid, or also bases, such as ammonium hydroxide. And this is one thing that happens within the digestive system. Remembering that the stomach is an acidic environment, as proteins are ingested and pass into the stomach, part of the role that the acid in the stomach plays is in disrupting the three-dimensional structures of proteins so that ultimately when those proteins that have been denatured come into contact with digestive enzymes, the digestive enzymes can work effectively on those unfolded, denatured proteins. A third way that proteins can be denatured is through the action of alcohols. And that is one reason why hand sanitizers are effective. When hand sanitizers come into contact with bacterial cells and penetrate the bacterial cells effectively, they ultimately are able to denature the proteins within the bacterial cell. The alcohol is not as effective at penetrating human cells, and hence, when one uses hand sanitizer such as this on their hands, it's able to target the bacterial proteins by effectively entering the bacteria cells and denaturing proteins there, rather than entering our cells and denaturing our proteins in a massive way. So now that we've taken a look at 3 prevalent ways that proteins are denatured, we are ready to dig into the next video where we'll be looking at chemical reactions that break the peptide bonds linking individual amino acids to ultimately release the individual amino acids for use by the body.